video we're going to think about the molecules that cells are made of. So what are their fundamental building blocks, which you may have heard referred to as the cellular macromolecules. So we're going to think about 4 different classes of cellular molecules. We're going to think about carbohydrates, we're going to think about lipids, we're going to think about nucleic acids, and we're going to think about proteins. So I'm just going to divide my paper up into 4 zones. Okay, so let's start with the carbohydrates. Okay. So carbohydrates, they have roles in energy storage, and they have a sort of structural role. So some carbohydrates, for example, cellulose in the cell wall of a plant cell is there to provide sort of mechanical strength. Okay, so let's think about the structure of a carbohydrate. Well, what we have when we're thinking about macromolecule structure is we need to think about monomers that get converted into polymers. So monomers are individual molecules, and polymers are long chains. Okay, so we might have our monomer, I'll just do them as circles. So we've got our individual molecules, so they're currently free from each other, okay? But when they get formed into a polymer, they then get attached to each other in these long chains. So there might be a linear chain, or in some cases you might have what we call branching. So I'll just annotate that there. So for some of them, that's a branch point. Okay, but we end up with them physically attached to each other. And in nearly all cases in biology, we have what's called a condensation reaction. So when we go from individual molecules, excuse me, to a polymer, we release a molecule of water. Okay, so we need to think about the monomers and then how they form the polymers. Okay, so for carbohydrates, your monomer is a sugar, sometimes known as a monosaccharide. Okay, so an example of a monosaccharide, there are others, but we might be thinking about glucose. Okay, so what's the structure of glucose look like? Well, glucose, we have a 6-membered ring with an oxygen in it. Okay, CH2OH, OH. So each OH is a hydroxyl. And they're pointing up different ones there. So if you remember, and the way that chemical structure are formed, anytime we've got these lines coming together, that means there's a carbon at the intersection of these lines. And in fact, we've got 6 carbons in a sugar. So we've got carbon 1, 2, 3, 4, 5, and then that carbon up there that's sticking out from the main molecule. Okay, so we've got glucose. We could also have fructose, which has a slightly different structure. It's still 6 carbons, but it's a 5-membered ring. The sugars are only made up of carbon, hydrogen, and oxygen. That's as complicated as their chemistry gets. Okay, so this is our monosaccharide. This is our individual molecule. Okay, they then get built up to form long chains. Okay, so we can then have, for example, we can have glycogen, or we can have cellulose. The different combinations of the glucose make it slightly different. So you might know that there are 2 slightly different forms of glucose. There's one called alpha and one called beta. The combination of the monomers you've got determines the overall structure. Okay, so for glycogen, for example, which is a storage carbohydrate in animals, what you've got there there's your glucoses. We have a bond with an oxygen between them. Okay, and we form structures like that. And in glycogen we can have branching, so we can also have another sugar up there, for example. Off it goes. So we have quite an elaborate structure of all of these individual glucose molecules. They're all bound together with a condensation reaction, and that bond there should be an oxygen with each one. That bond between them we call a glycosidic bond. Okay, so in this case we've got what we call a 1,4 glycosylic bond. So this is what we call an alpha glucose, and these are all alpha glucoses. So this is in fact an alpha 1,4 bond, and we can have other different bonds. So apologies, I've shown that in slightly the wrong place. I'm going to get rid of that there. So we can also have coming from there, we can have an oxygen going up there. So we can have these branch points in there. So we've got our glycogen, we've got our alpha glucoses. They're all connected together with these glycosidic bonds. They form these elaborated structures. Okay. So that's a brief overview to carbohydrates. There's obviously more complexity of carbohydrate structure than I've shown you there, but that's the overall plan with carbohydrates. Now I'm going to do lipids. Now lipids are a little bit different. They don't follow this monomer-polymer structure. So lipids are mostly to do with membranes. They also have some roles in signalling. Okay. So lipids work a little bit differently. They don't work in this monomer-polymer bit. They work like this. So an individual lipid, so we might think about phospholipids, which are the ones that form the membrane. Okay, so for a phospholipid, the structure is like this. We've got 3 carbons as the sort of central group, and that's a group that we call glycerol. So 3 carbons. One of those carbons is attached to a phosphate group. So there is some oxygens in there as well. I'm just not showing the entirety of the chemical structure. We've got a phosphate group there, and then via a bond with some oxygen, we've then got long fatty acid tails. So these are just CH2, 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 so they're just carbon and hydrogen. Okay. So that's the overall structure. There are a couple more atoms in there. I'm not going to go through each and every atom, but that's kind of roughly the structure that we're thinking about. And when lipids come together, they don't form these kind of covalent bonds between each other. They actually just sort of sit next to each other. So if we think about the structure of the membrane, then we often draw the membrane like this. So we have a circle and 2 little lines. You'll have seen this in textbooks before. And I've got a whole video on membrane structure. You might then have a protein in there, but then you've got more of these little molecules like this. Okay, we'll ignore the protein for the moment. We're just thinking about the lipids. So each of those individual diagrams is one of these phospholipids. And the way that these diagrams work is this bit is the head group, which is a polar part of the molecule. So that is hydrophilic. It will like interacting with water. So the polar head groups are these groups here. Okay, so that means the glycerol and the phosphate. Okay. Whereas the fatty acid tails down here, okay, so the tails are non-polar, so they will be hydrophobic. Okay, so the polar bit, the non-polar bit, sorry, is in the middle. Okay, and then we've got, again, we've got those head groups on the outside. Okay. So these lipids haven't actually physically attached to each other. They're free to move. They can move, you know, this lipid might move over here, might go elsewhere. They're not physically attached to each other, but they kind of stick together in the membrane like this, so that we've got this non-polar hydrophobic bit in the middle of the membrane. So effectively we've got a layer of fat in the middle of the membrane, and then we've got these phosphates groups on the outside that are interacting with the water. And that's what allows the membrane to have its structure. So lipids aren't really a true macromolecule. They're sort of more an association of macromolecules because they're not physically bound to each other. This doesn't undergo any reaction when it goes into the membrane. It's just sitting next to them. So it's a little bit different to the monomers polymers thing. Okay, let's get to proteins, everyone's favourite. So proteins have so many different roles. They can be structural, they can be enzymes, they can be receptors. Basically anything, almost anything interesting that's happening inside a cell is almost certainly catalysed by a protein. Okay? So proteins do follow this monomer polymer thing. So the monomer is what we call an amino acid, which has the structure like this. So we've got nitrogen. So we've got an amino group. So we've got, so here we've got an amino group. That's what gives it its name. We've got a central carbon. Okay, notice that carbon is bonded to 4 different things, and we sometimes call that the alpha carbon. We've got a carboxylic acid group at the other end, so C double bond O and OH. We've got a lone hydrogen, and then we've got this bit called R that you may have seen before. So we've got an R group. Okay. What that R group means is this is different for every amino acid. Okay, so there are 20, 21 naturally occurring amino acids, depends on whose definition you take. Each of those has a different R group. Okay, so for example, for the amino acid glycine, the structure would be, I'm just going to summarise it, H2N, carbon, COH. For glycine, we've just got a single hydrogen there. Okay. But for other amino acids, then you'd have more complicated structures. So it might be that for another amino acid, then your R, so that's for glycine. It could be that R equals CH2, CH3, for example, okay, would be a different amino acid. Apologies, I can't remember the name of which amino acid that is off the top of my head. Okay, so different amino acids have different R groups on the side of them, and that affects their properties when they come in and form a protein. Okay, so this is our monomer. So R is different for each different amino acid. Okay. So these then come together and form a polymer, and when they do, it looks like this. Okay, so we have N, C, C double bond O, N, H, C, C, N. So that would go that way, that would go that way, so on and so forth. Okay, so we'd have. Then we'd have our R group sticking out the top on that one. It's sticking out the bottom, sticking out the top. Okay. There's the H. There's the H. Okay. So this is now our long polymer. Okay. And we can see that we've got a characteristic bond keeps forming. So this in here, and this in here, are what we call a peptide bond. Okay, so every time an amino acid links together, we've got C double bond O, N, H, that's our peptide bond. Okay, and then you notice that the R groups, which are the most important things really for thinking about the function of the protein, stick out either side each time. Okay, so now those R groups are sticking there. So we've then got the beginnings of protein structure. So this is what we sometimes refer to as the primary structure of the protein. Okay, so we don't have any branching in protein. So it is literally this model of a polymer. Okay, so if each of those is your amino acid. But what we know with proteins is that they can then fold in weird and wonderful ways. So I'm just going to do this. You know, I'm not going to go through the entirety of protein structure here, but what we've got is then we've got a complicated three-dimensional structure. So that backbone, each different one is a different amino acid with a different R group. It folds up. And there'll be another video about protein structure for you to have a look at. Okay, so we start with a monomer, which is an amino acid. We then go to a polymer, which is this chain of amino acid links by peptide bonds. We've got the R groups pointing out in different directions, and then we sort of then build up to more elaborate structures. Okay, so the next thing that we need to think about is nucleic acid. Our final major form of macromolecule, and here we're thinking about DNA and also RNA. Okay, so let's have a think about these, because these are obviously the backbones of our genome. Is made up of our genome is made up of DNA. Okay, so that's our final class of macromolecule that we need to think about. Okay, so again, let's build it up with the monomer. Is a nucleotide. So which has a structure that looks like this. So we have what's called a ribose sugar. So we have a 5-membered ring. Okay, we have a phosphate group, and then we have what's called a base. Okay, and the bases have a bit more elaborate structure. So I'm just going to draw in there. There's some double bonds like that. Might be some NH2. There's quite a lot of nitrogen in these. Okay, so if we just label the different bits of this, okay, so that is a ribose sugar. We have a phosphate, and we have a base. Okay, that ribose sugar, if it is in RNA, we'll have hydroxyls on both there. If it's in DNA, then it will have an OH there and just an H on the corner there. Okay, so this is for RNA, this is for DNA. So it's a slightly different sugar there. But the most important thing is we've got the sugar, we've got the phosphate, and we've got a base. And you probably know that the bases for DNA are A, T, C and G. So adenine, thymine, cytosine and guanine. So this is adenine that I've shown. So it's a particular combination of rings and nitrates and whatever. Each of those is a slightly different structure. And then we have RNA has the bases of A, U, C and G. So RNA doesn't have T, it has U instead. So how do these get linked together? Okay, so notice that this is a much more complicated molecule than anything we've had before. Okay, but the way these get put together is like this. Okay, so if we start with our little ribose, okay, and then we've got off there, we're then going to have the phosphate bound to the next ribose like this. So you sort of have sugar, then phosphate, sugar, then phosphate, sugar, then phosphate. Okay, so that is known as the sugar phosphate backbone. Okay, so you've got sugars and phosphates. That's the same all the way along, but then we've got a different base going on. So I'm just going to draw these as rectangles. So those are sticking out. Okay, so that might be an A, that might be a C, that might be a G, for example. Okay, I'll just do those again: A, C, and G. Okay. So in RNA is usually single-stranded, so you just have it like that. But DNA, we probably know, is a double-stranded molecule. So the way that DNA works is like this. So DNA goes the opposite direction. This is a little bit complicated to draw. So then we've got phosphates, then we've got same thing again, and a phosphate. So we've got another sugar phosphate backbone, like that, but notice this is going in this direction, this is going in this direction, okay? And then we can have the complementary base sticking out, and that can form bonds between them, okay? So you might know that A always pairs with T, C pairs with G, so G pairs with C. Okay, so those are our bases. So they are forming what we call hydrogen bonds. So they're not covalent bonds, they're just little hydrogen bonds. But that's how we start to get the double-stranded DNA molecule. So we've got the sugar phosphate backbone, and then in the middle, we've got the base pairs. And that underpins the structure and function of DNA. So I've whizzed through quite a lot of just fundamental building block stuff here, okay? I've not given all the detail, I've not given all the chemistry of it. Obviously for each of those, there's more detail that's there, and if you really want to get into the biochemistry, you can do, but hopefully that's helped you to just understand the fundamental building blocks. So most of them, we've got monomers get converted into polymers by sort of being linked together in chains. We have carbohydrates that are based on sugars. We have proteins that are based on amino acids, and we have nucleic acids that are based on nucleotides. And slightly separately, they don't follow the same monomer polar rule, are the lipids, which are involved in the membrane. Okay, so have a look at that. You might want to look at a biochemistry textbook or a cell biology textbook to help you with that. As I say, I've gone through quite a lot of stuff